I'm really excited for my 3004-3004H bio class, but I don't know where anything is. Can you guys show me?
Of course.
Alright, let's see. So every group in this class will be assigned a lab bench, as indicated by your number here. So I was group E1. This is our lab bench. And there's a number of things that are really important to know about in your bench. Every bench will have outlets here for your laptops or whatever. You'll have this space, as well as this tape will indicate where you can put your saved plates. So if you have plates that you want to save for your bacteria, you can store them here. As well, every group will have a tray with kind of the essentials for every procedure that you would do. You'll see here we have pipette tips in the orange and blue box. The blue box is for the larger pipette tips, the orange is for the smaller ones. You also have a test tube rack, as well as a microcentrifuge rack.
Okay, and then you have the ethanol here for when you have to sterilize things, and then you'll have also a Sharpie to label things or just to write notes on here, and then here's a little notepad. In addition to this, there should be, between a couple lab benches, the ethanol and the distilled water will be shared.
Oh man, I really want to make my culture tubes, but I don't know where the pipettes are. Can you help me?
Yeah, let me show you where they are.
Sweet.
So all the sizes of pipettes are over here, like the main ones. You've got the 100 to 1,000, the 20 to 200, and the 20 to 20, 2 to 20 microliters, and they're all also labeled by color. And then down here, there's also safety glasses in case you're using HCl or general electric bases or something like that. And if you need smaller ones, there are pipettes here that are pink that are 0.1 to 2 microliters as well.
Let's see, and over here on the other side of the lab, we've got a number of things. We have a lot of our larger pipettes for our serological pipettes when you're working with larger volumes. We've got like 5, 10, 25 milliliters. Also, we have all this stuff for Bunsen burners. So if you're going to be using dilution plates or just general plating, you have Bunsen burners here, as well as the loops that'll spark them. And let's see, and that's about it. All right, let's see. Welcome to the incubator room. And here you'll find these 3 incubators are pretty much the 3 that you would use if you were incubating your plates. And there's no difference between them other than their labels. So just remember which ones you put your plates into. One really important thing about these is to make sure that you're not putting tubes in, as well as that you don't keep them in for an extended period of time. Otherwise, you might lose cleanup points. If you do have them in for a longer period of time, then the TAs will notify you, and you got to make sure to check on them so you don't lose points. Additionally, down here, we have our shaking culture. So if you're ever working with shaking cultures, what you're going to do is you're just going to put your tubes in there, you're going to date them, and then you can get them whenever you need them, usually for about 24 hours.
Okay, so then in here, there are the fridges for your media and any plates or other things that you'd like to keep cool so they don't grow further. They're labeled by evolution section, so just stay on the right level and you should be fine. And then over here is the freezer in which they keep the frozen stocks, and as you can see, students aren't allowed to touch this, so you'll just need a TA for help.
So after you're done doing your procedure, whatever you're making, if you're making plates or tubes or cultures, anything that touched bacteria that could have been contaminated, what you're going to do is you're going to put it in these red bins that are all around the lab. This is the autoclave waste, so where it's going to go in order to be sanitized so you can use them again. That includes pipette tips, that includes culture tubes, but there's not going to be any glass. So if anything is glass, it'll go in Kill Cycle or somewhere else. All right, and if you have any other questions, you can always go to any number of TAs that are around the lab. You can tell who they are because they've got the nice little name tags. Let's see.
Hi, I'm Lyric. I'm a 3004 TA, and I'm here to answer any questions you have when in lab.
All right, and if you're working with cultures, which most of you should be, what you're going to be doing is you're going to be using lots of different tubes, and at the end of their lifespan, they're going to need to go in the autoclave. But however, they're not going to go into the autoclave red waste bins. They're going to go into the Kill Cycle. So you're going to put them in here, and you want to make sure to take the tape off, or else you might lose cleanup points. And you're just going to put them here, while they'll be autoclaved and returned for later use.
Okay, so when you need to make media or plates, this would be kind of the area in which you'll do it. Most of what you're going to use is going to be in these types of containers, and they'll all be labeled autoclaved prior. Over here, there's going to be the weigh books and the scoopulars and whatnot. And then these are the scales. And then a lot of what you're going to be using is going to be in these cabinets. For example, protease peptone to make the media and the plates is right here. Alright, so then there are more supplies over here, but the ones that will be most relevant are glass spreaders for when you're making plates or culturing plates. And then if you are going to be using chemicals, there's some under this flame hood. And then whilst you're using them, you should also wear proper protective equipment.
Right, so one of the most important things about labs is actually making sure that you've cleaned up once you've left. You don't want to leave any kind of mess on the table. That means when you're done, you're going to wipe it down with ethanol to sterilize it. You're going to take any cultures or tubes, you're going to put them into proper disposal. And so another thing would be, let's say you put some plates in the incubator, you don't want to leave them in too long. Here we have an example of what might happen. This group left their plates in the incubator for too long, and as a result they've lost some points, which will be indicated by this kind of notice. So you want to be aware of that.
Wow, thank you. Now I finally know where to find everything in this lab. I'm finally ready for Bio 3004/3004H.
I'm so excited. Yeah.
Yeah.